And this is the second second half of the PowerPoint DNA structure and replication. This video lecture is going to be on the replication portion only. So when we're looking at DNA replication, we have talked about this before when we've gone through mitosis and meiosis. And we will start with a original and end up with two new strands. And the animation to the right is going to kind of show you the big ideas about it. We're going to follow those base pairing rules that we learned when we looked at the DNA structure. First question is, when does it replicate? And if you reflect back to what we learned about mitosis and meiosis, you remember about the cell cycle and interphase having G1, S, and G2. During the S phase is when I had discussed about how the chromatids duplicated to create the chromosomes, and the S phase of the cell cycle. It also replicates again in meiosis too. So the other question is, why does it replicate? Because the end product of mitosis and meiosis is going to need those chromatids separated into the different new cells. There are a lot of enzymes that are involved in the process of DNA replication. And the first thing, we need to go back to what we learned in the fall semester about enzymes. An enzyme is a molecule that makes a process work, and they can speed up a process to occur in cells. In this particular case, these enzymes have to be there for this process to work. And it's really important for this to occur at the right pacing, at the right process, so that the mitosis and meiosis can work appropriately. There are a lot of enzymes when it comes to replication. When I show you some of the other videos that are on YouTube that are published by other scientific people that you're going to watch, you're going to hear about a lot of other enzymes, but the two enzymes that I'm going to hold you required to know is helicase and polymerase. So just be aware that other things are going to be discussed, but helicase and polymerase is going to be the two main ones that you need to know. This little animation at the bottom shows you the helicase. And what I want you to remember about that is hela and hydrogen go together. Helicase, hydrogen bonds. They break them apart. You could also think about it as like, if you see it, it kind of looks like the word helicopter. And so I want you to think about like the blades of the helicopter kind of going through there and chopping those hydrogen bonds apart. So they're breaking it and opening it up. The second enzyme is going to be called the polymerase, DNA polymerase. And they move along the DNA strands and they add nucleotides to the exposed nitrogen bases according to the base pairing rules A to T and C to G. And this is another animation that is pretty interesting, and it does show the helicase breaking the hydrogen bonds. And then the green characters are going to be the polymerases. So think about them as little workers that are going along and adding puzzle pieces, the correct nucleotides to the matching one, according to the A to T, C to G base pairing rules. These are a couple video clips I will put and add to this posting on Classroom. So let's go through some of the steps when it comes to the DNA replication. The step of DNA replication is that the DNA double helix will unwind, or the DNA double helix will unwind. The DNA helicase will go along and break the hydrogen bonds that link the nitrogen bases. In the third step, DNA polymerases move along each of the DNA strands and add nucleotides to the nitrogen bases. The polymerase follows the base pairing rules of A and T go together and C and G go together. This is a big picture of this, and you can see that where this is the helicase in the middle, so it's splitting it in half. This is also called a replication fork. There are also these free nucleotides that are floating around. And the polymerase will move along this strand and will add A to T, C to G. In some of the videos I'm going to show you are going to talk about how in one part of the strand it's going to be called the leading strand. And because the arrangement of the nucleotides is in one order, the polymerase will continue on in that pattern and add straight up. It's just following the helicase. But if you remember when we did the DNA model in class and you colored and cut out the nucleotides, on this side at the very bottom, this is going to be called the lagging strand. They're actually turned upside down and facing a different direction. So the polymerases have to add them in the same direction, the same way. They can't just turn it themselves upside down and go in a different direction. So it's going to be called the lagging strand. They're going to be pulling these apart and breaking them into portions. Each one of those portions is going to be called an Okazaki fragment. Knowing the difference between a leading and lagging strand and Okazaki fragment is not necessary for your assessments. In the end, you will have two end products that are two DNA strands, and they're going to be the same genetic material with the same functions. And these video clips that are tagged in this PowerPoint, you can access them on the PowerPoint, or I will be adding them as an external link as well.